So in this lecture, we're going to spend our time talking about lysogeny and about what we call temperate bacteriophage. Temperate bacteriophage is the name that is more often used to describe the bacteriophage that have both of these life cycles, both the lysogenic and the lytic life cycle. And this is how you spell temperate. T-E-M-P-E-R-A-T-E. Before we get into this conversation, though, there are a couple of things that I need to bring up from the past couple of lectures that I wanted to just make clarifications of. The first is I wanted to go back to the genome of T4 and T7 and just point out a couple of things. First, most of what we talked about with respect to the lytic life cycle of bacteriophages was in relationship to T4. The second thing I wanted to note is that the T4 genome, remember that it will undergo circularization once it's entered into the host cell, and that's because it has this terminal redundancy at the end, because what the bacteriophage has done is encapsulated into its capsid a full genome plus a little extra genomic material that is a duplicate of what's on the other end. T7 bacteriophage, though, have a linear genome as well, but it does not circularize. So T7's life cycle is a little bit different than that of T4. And so the other thing to point out here is that T7, unlike T4, actually encodes its own RNA polymerase. And it encodes its own DNA polymerase as well, shown here. But remember, this is in contrast to T4. T4 uses the host DNA-dependent RNA polymerase for its transcription, and it transitions from immediate early to late gene expression based on what is that DNA-dependent RNA polymerase binding to. So what promoter regions is it binding to? And that in part can be controlled by things like GP55, which is going to allow for the RNA polymerase from the host to identify late promoters. Now, the one other thing that I did want to mention, as I'm moving ahead here, I'm actually going to go back. There are a couple of things, again, that are required for gene expression in T4, and that includes a transition from using the host sigma factor to recognize receptors to ones where eventually GP55 completely replaces the host sigma factor to recognize those late promoters. One additional thing that needs to take place is actually replication needs to begin before those late genes can be expressed. And so in order for those structural proteins to be expressed or be transcribed, you must have DNA replication occurring as well. So in this lecture then, what we're going to cover is lysogeny, this part of the viral life cycle that is kind of analogous to latency in eukaryotic viruses, and in some ways analogous to retroviruses as well. What we find in latency, the decision of whether a temperate bacteriophage undergoes lytic or the lysogenic life cycle is going to be dependent on a couple of factors, and we'll talk about these factors towards the end of this lecture. But let me just introduce you to a couple of the brief steps. There has to be a decision made, and that is what we'll spend most of our time talking about today. Temperate bacteriophage will integrate their genome into the host genome as what we call a prophage. And these prophage can make their way back out if some sort of stress or signal triggers for the virus to reactivate lytic life cycle. What we can find as well, though, is that the prophage undergoes some mutations, preventing it from ever being able to produce infectious virions. And so we call these cryptic phage. Now there are some characteristic proteins that we will find that control lysogeny. The major one that is expressed only during lysogeny, sorry about that, is a protein known as control 1 or C1. During true lysogeny, this is the only protein that is being expressed. There are other ones that are contributing to the quote-unquote decision to enter lytic or lysogenic life cycle. And those include the proteins CRO, that includes the protein N, and it includes a couple of other proteins shown in the genome to the right here, C3 and C2. So what I'm going to spend the time talking about today is how do these proteins play a role in determining whether a cell undergoes lytic or lysogenic life cycle. In brief, the simplest way to explain it is that there's a competition between CRO and C1, and these two proteins antagonize each other. More specifically, they inhibit each other's transcription. And so in doing so, these are the two major proteins that contribute to this decision. Now we'll be talking about this process with respect to a phage known as lambda phage, which infects E. coli. And the first thing that happens during lambda phage infection is transcription from a leftward promoter known as PL1 and a rightward promoter known as PR. I'm not worried with you knowing these names, but I'm just trying to point out that these two promoters will drive the transcription of different set of genes. PR will drive the expression of CRO. And so from this PR promoter, we will find that there will be some gene expression, and transcription will stop at a terminator sequence. In the same way, N transcription is controlled by PL1. Transcription will proceed until there is a termination signal. Now, the function of N is to bind to a host protein known as NUS1. And when N and NUS1 bind together, they allow for the host RNA polymerase to actually continue reading past these termination signals that exist in both the CRO and the N mRNA regions. And so when N binds to NUS-A, and when these bind to RNA polymerase, transcription will proceed further. And so from the rightward transcript, we get both CRO and production of C2. And from the leftward promoter, we get transcription of N and C3. And that takes us to the next step of the process. What is the role of these two proteins? Okay, so what I've told you so far is early on in this transcription, we've got CRO being expressed, we've got N being expressed, and when N binds to NUS-A, these will help drive the transcription of bicistronic mRNAs, ones that encode both CRO and C2, or ones that encode N and C3. The function of C2 and C3 is to drive the expression or to activate transcription of C1. C3 is a stabilizing protein. Its function is to stabilize C2 and to prevent its proteolytic cleavage or its degradation. We'll talk about how this happens a little bit later on. C2 will recruit RNA polymerase to a promoter that will drive the transcription of C1. So in order for C1 to be expressed, C2 is necessary and required. In the next step of this process then, we're going to take a look at the promoter region for C1 and for Crow and talk about how these two mutually antagonize each other's expression. So here is a rather complex picture showing the interaction between both C1 and Crow at two different promoters, which we call PR and PRM. PRM controls the expression of C1. PR controls the expression, as we said before, of Crow. The other thing to point out here is that there are three operator regions between these two promoters, and these operator regions are shared. And these are known as OR3, OR2, and OR1. Now, what I'm going to start out by saying is that both Crow and C1 will bind to these three OR regions. And so in other words, the two of them are competing with each other for binding sites. They bind to these three different regions with different affinity. And the importance is not so much to remember the order, but to remember then what that means is that these three sites are going to have slight deviations in their nucleotide sequence in comparison to one another. Now, Crow binds to the ORs in the following order. It first binds to OR1, then OR2, and OR3. And when Crow binds to OR3, this functions as a transcriptional blockade. Binding to OR3 will prevent transcription of C1 from occurring. At the same time, the polymerase is able to bind to the PR, or that rightward promoter, and to drive transcription of Crow. In this case, then, what we find is the cell will undergo a lytic life cycle, starting to express genes that are involved in DNA replication and lysis, and the cell will be blocked from going into, or the virus will be blocked from going into lysogeny. In the other case, what we find is that C1 will bind to OR1, 2, and 3. It binds to OR1 the weakest and OR3 the strongest. And when this particular C1 protein binds to OR1 and OR3, we find the following. OR1 and OR2. C1 first, these binding sites will inhibit the transcription of Crow. At the same time, we find that actually the binding of C1 to OR2 will activate more C1 expression. And so we have not only a negative feedback, but a positive feedback here, where C1 is driving more of its own expression. Transcription in this direction, then, will lead to lysogeny and block the lytic life cycle. Now, the process of lysogeny is reversible, meaning that if a viral genome remains intact, if there are not mutations that affect the expression of proteins like Crow, then we can find a reactivation of lytic replication. The point to make here, though, is that lysogeny exists in what we would consider to be healthy cells or unstressed cells. And so what we're going to talk about here are two different factors that contribute to the decision of whether a virus, whether a temperate phage will enter lysogeny or the lytic life cycle. First, to establish lysogeny, the cell has to have a lot of energy present. Low amounts of energy will actually prevent lysogeny from being established in the first place. So some of you may remember ATP is the primary quote-unquote energy currency of a cell. And when energy is being used in the form of release of phosphates, ATP gets converted into ADP, and the release of another phosphate will form AMP, and eventually AMP undergoes a reaction in which it circularizes, driven by the enzyme adenylyl cyclase, to form cyclic AMP. Cyclic AMP is what's sometimes described as an alarmone, meaning the expression of it, or its presence, is an alarm to the cell that energy levels are low. What we find in this case here is that cyclic AMP will bind and activate a protein known as HFLA. Well, HFLA is a protease, and one of its targets is C2. So what we find is that the activity of HFLA will increase as cyclic AMP binds to it, and it will overcome the stabilizing forces that C3 has on C2. Without C2 present, we will find no transcription of C1, and therefore lysogeny can never be achieved in the first place. Now let's assume that the virus has undergone integration into the host genome. We have a prophage, and then there is a period of time of stress. And most often stress is going to come in the form of those that lead to genomic instability or DNA damage. So these would include things like UV light, hydrogen peroxide, and even some chemical treatments like a drug known as mitomycin C, which is known to cause DNA damage. You could consider ethidium bromide possibly to be another example as well. In these cases, genomic instability is recognized and leads to the stabilization of a protein known as REC-A. REC-A stands for recombination A, and it's required for protecting E. coli and other bacterial cells against genomic instability. It's part of what we call the SOS system. Well, REC-A is a protease as well, and it can cleave C1. And so when DNA damage is detected, REC-A expression or levels increase because it is stabilized, and that will lead to its ability to degrade C1. If we take a look back at our promoter, what we find is the following. Degradation of C1 would remove C1 from both the OR region. That now leads to a loss in transcription of C1, and it leads to a loss in the ability to inhibit CRO expression. And so, by this process, we would find that DNA damage increases the expression of the CRO mRNA and the mRNA encoding the CRO. CRO protein expression increases, and now we find further inhibition of C1 and transcription of those genes that are required for the lytic life cycle. So one of the things that we can think about is, what is the evolutionary benefit to lysogeny? And what is the benefit compared to the lytic life cycle? Both of these mechanisms exist because there's been selection for them. And we've seen here some of the cues that can lead to the establishment of lytic or lysogenic life cycle. It should make some pretty clear sense the benefit of the lytic life cycle with respect to evolution. The goal of evolution is to ensure that genetic information is passed along from one generation to the next. And in the case of the lytic life cycle, the virus gets into the host, replicates rapidly, and then eventually produces a large number of progeny viral particles that are capable of infecting more cells. In lysogeny, the process is a little bit different. In lysogeny, the viral genome is integrated into the host genome using an integrase or a transposase. And each time the host cell divides, the viral genome is replicated along with it. So why is this beneficial, and why is the lytic life cycle beneficial? Or when might there be a benefit of one over the other? Well, the lytic life cycle is particularly beneficial in periods where we have high concentrations of the host cell. For example, think of the gastrointestinal tract in E. coli. Concentrations of E. coli in the gastrointestinal tract are abundant, and so it might be quite beneficial for lytic phage to also gain access to this bacteria as well, because there's an unlimited supply, theoretically, of the host. But what if the host is more rare? Or what if this is a condition in which there is no place for these progeny virion to go after they've performed their lytic replication? That's the benefit of lysogeny. In lysogeny, as the host replicates, the viral genome replicates as well. And so the virus doesn't need to be producing new viral particles in order to ensure that its genomic information is passed from one generation to the next. Now what this image shows is that it's as if all of these cells undergo conversion from lysogenic to the lytic cycle at all the same time, and that's not really what happens. Think about our daily interaction with bacteria. For example, I've been touching the surface of the iPad which I'm recording this on for the past 20 minutes. I'll take that bacteria from the iPad and it's going to spread to other areas that I touch throughout the day. For example, my desk, my coffee mug, and even at home. And as we move bacteria from one location to another, this allows for the dissemination of not only the bacteria, but also the lysogenic bacteria, or the lysogenic virus that may be inside that bacteria as well. So it allows for a spread of that microbe, potentially over much greater differences than we may find with the lytic phage. Finally, we describe sometimes the process of bacteria gaining one of these lysogenic phage or temperate phage as lysogenic conversion. And what we find is that lysogenic conversion can improve the quote-unquote hardiness of a bacteria. It can also contribute to pathogenicity. There are a couple examples of how this can happen. In some cases, viruses make mistakes. They not only package the host genome, but they may, upon removal of their prophage from the host cell, may actually grab or accidentally take some of the host DNA with it. So instead of cutting at those locations where the viral genome is, they might cut and include a little bit of the host DNA as well. Well, in these cases then, the host DNA would become part of the viral genome. And if that host DNA includes toxins, resistance markers, other virulence or pathogenicity factors, these can be spread from one bacterium to another through horizontal gene transfer. There are cases in which we find that temperate phage can include factors that improve the hardiness of bacteria. For example, Bacillus has an improved rate of sporulation with the presence of particular temperate bacteriophages. And Bacillus anthracis has the ability to form biofilms because of lysogenic phage that are now stably part of the bacterial genome. We see other examples as well in E. coli, where a bacteriophage known as H19B, which is now no longer produces at great frequencies lytic virus, but is largely stable within the genome. It produces what's known as STX1 and 2, which are Shiga toxins. Vibrio cholerae has a bacteriophage known as CTX, which has in its genome the cholera toxin. And so what we bring up here is that bacteriophage can increase not only fitness of bacteria, but they also can increase the toxicity or the pathogenicity of some of these microorganisms. And this is some of the downside of bacteriophage. So if we go back to the general life cycle, we have covered now not only the lytic life cycle and how this contributes to bacteriophage getting in and out quite quickly and producing mass numbers of viral particles, potentially on the range of 100 per one infection. And we've talked about lysogeny and the role of temperate phage in establishing lysogeny, how that lysogeny is established at a molecular level, and we've talked a bit about lysogenic conversion and the benefit or evolutionary benefit of lysogeny and lytic replication.